Welcome to our exploration of how psychological stress can accelerate immune aging, a topic gaining attention in medical science. Recent research reveals that stress doesn't just affect our mood, it can actually speed up aging-like changes in our immune system. This process is closely linked to the gut microbiota, the community of microbes living in our intestines. Today, we'll break down the science behind this connection, starting with how stress impacts the body, then moving through the brain-gut-bone-marrow axis, and finally discussing what this means for healthy aging. Let's dive into the fascinating world where mental health and immunity intersect. Psychological stress is increasingly recognized as a risk factor for several health conditions, including cardiovascular disease and diabetes. When stress is paired with a weakened immune response, the risk becomes even greater. Scientists are now focusing on how chronic stress can influence the body's defense mechanisms, making us more susceptible to illness and age-related diseases. Understanding this risk is crucial for both prevention and intervention as it highlights the importance of managing stress for overall health. Let's look at how stress signals travel through the body and impact our immune system. The heart of immune aging are hematopoietic stem cells, which reside in the bone marrow. These cells are responsible for producing new blood and immune cells throughout our lives. A study published in Cell Stem Cell shows that psychological stress speeds up aging-like changes in these stem cells. This means that under chronic stress, our body's ability to generate fresh immune cells diminishes, making us more vulnerable to infections and diseases. The connection between stress and stem cell function is a key piece of the puzzle in understanding immune aging. Brain-gut-bone marrow axis is a newly discovered pathway that explains how psychological stress affects immune aging. Stress-responsive regions in the brain regulate the balance of gut microbiota, which in turn influences the function of hematopoietic stem cells in the bone marrow. This axis shows that mental health and gut health are deeply interconnected, and disruptions in one can lead to changes in the other. By understanding this communication network, researchers hope to develop new strategies to protect immune function during stressful times. Chronic stress activates the body's fight-or-flight response, which is controlled by adrenergic receptors and inflammatory pathways. This response is designed to help us survive immediate threats, but when it becomes chronic, it can disrupt immune function. Previous studies have shown that these stress signals influence the formation of immune cells in the bone marrow, but the exact transmission route was unclear until now. The new research sheds light on how the brain communicates with the bone marrow through the gut microbiota. Researchers used 4 different mouse models of stress to study the interactions among the brain, intestine, and bone marrow. These models allowed scientists to observe how chronic stress affects various physiological systems. They found that stress reduced activity in 2 key brain regions, the medial prefrontal cortex and the periaqueductal gray. This reduction led to loss of hematopoietic stem cells and decreased production of lymphocytes, the cells that help fight infection. The findings highlight the complexity of stress's impact on the immune system. One of the most significant discoveries was the change in gut microbiota in stressed mice. Specifically, there was a loss of Lactobacillus reuteri, a beneficial species that helps maintain a healthy balance of gut microbes. This loss was accompanied by lowered levels of spermidine, a compound crucial for clearing out damaged cells. These changes in the gut microbiota played a central role in mediating communication between the brain and bone marrow, ultimately affecting immune function. Suppression of just 2 specific brain regions, the medial prefrontal cortex and the periaqueductal gray, was enough to produce many of the immune defects caused by psychological stress. This finding was surprising and suggests that targeted interventions in these brain areas could help mitigate immune aging. By focusing on how the brain regulates gut microbiota, researchers are opening new doors for therapies that support immune health during stressful periods. Spermidine, a naturally occurring compound, was found to play a crucial role in clearing out damaged cells and maintaining immune health. In stressed mice, levels of spermidine dropped, which contributed to immune dysfunction. This highlights the importance of microbial metabolites in the communication between the brain and bone marrow. Understanding how spermidine and other compounds influence immunity could lead to new ways to support healthy aging. While the study provides valuable insights, several important questions remain. How does psychological stress alter neural circuits in different disease settings? Are similar mechanisms at work in humans, or are they unique to mice? Researchers plan to investigate these questions further, aiming to understand the broader implications for human health. The answers could pave the way for new interventions that protect the immune system during chronic stress and aging. Researchers are also exploring whether interventions can be developed to improve bone marrow function in aging or during times of chronic stress. These interventions might target the gut microbiota or the brain regions involved in stress signaling. Although substantial work is needed before clinical translation, the findings provide a conceptual framework for developing new approaches to mitigate immune aging and stress-associated immune dysfunction. Managing psychological stress may not only improve mental well-being, but also help preserve immune function and promote healthy aging. This broader perspective encourages us to see stress management as a key part of maintaining both mind and body health. Simple lifestyle changes, mindfulness practices, and medical interventions could all play a role in supporting immunity and reducing the impact of stress on aging. This research was supported by several major programs, including the National Key Research and Development Program of China and the National Natural Science Foundation of China. Such support highlights the importance of studying the links between psychological stress, gut microbiota, and immune aging. Continued funding and collaboration will be essential for advancing our understanding and developing effective interventions. The recent studies have explored related topics, such as how blood protein clocks flag higher risks of death and chronic disease, or how cellular senescence interacts with stemness and disease. These findings contribute to a growing body of knowledge about the factors that influence aging and immune function. By connecting the dots between stress, gut health, and immunity, researchers are building a more complete picture of what it takes to age healthily. In summary, psychological stress can accelerate immune aging by altering the gut microbiota and affecting hematopoietic stem cells. The brain-gut-bone marrow axis is a key pathway in this process, and understanding it opens new possibilities for interventions. Managing stress, supporting gut health, and advancing research are all crucial steps toward promoting healthy aging. As science continues to unravel these connections, we move closer to a future where mental well-being and immune health are protected together.